and look happy. Okay, now I'm going to take a photo. 3, 2, 1. All right, done.
Okay, thank you all for joining from such diverse places. What we're going to do to start this is we are going to, I'm going to give an introduction to ocular melanocytic tumours, in particular pointing out the things that you might find difficult to notice when you're looking at slides. So I will now share my screen. And here we go. And I will now change it to slideshow. And you should be able to.
Yep, looks good.
That's great. Okay, so I'm going to ask Amanda to monitor any chat. So I'm open to interruptions, but if you feel a bit shy, please just write it in the chat and Amanda will interrupt for you.
I'm good at that.
Okay. So let's think about, in particular, we're going to think about uveal tumours. So a lot of information can be gained by looking at the low-power views, i.e. the macro and the whole slide view. So when we're reporting tumours, often we're asked to describe the shape of the tumour. So either dome-shaped or mushroom-shaped, or I would call this a nodule, and I would probably call this a mushroom too. So let's have a think about what gives the melanomas these shapes. If I see a dome like this, I can imply that it's still contained within Bruch's membrane, whereas this one here has broken through it. Another question that's often asked is how do we measure the base diameter? So we measure around here from the farthest we can see under the microscope, and the height I would measure from here through to here, the farthest height. And we measure that on all tumours. Other things that we will notice macroscopically that can help us stage the tumour are involvement of this is the involvement of structures that extend beyond the sclera or pigmentation on the scleral surface. So if you're asked what observations you would make macroscopically, those things. This one has got massive extension into the orbital fat here, but actually a relatively small tumour on the inside. And of course, these will all have correlates down the microscope. So here, for example, we're seeing a melanoma that's involving the optic nerve and extending into the extrascleral fat.
Diane, can you just go back and point out which bit is the optic nerve and where is the sclera?
All right. So good point. This is the
E.
This is the choroid here. This is Bruch's membrane here. This is the tumor that's broken through Bruch's membrane here. This is the optic nerve here. This is the gap between the optic nerve and the meninges. This is the sclera here. And this is the extrascleral fat.
Okay, so we're just looking at half of the optic nerve, really. It's sort of been cut almost down the middle. Yep. Okay,
so through Bruch's membrane, here we go. So everything that has a pinch in it is likely to be through Bruch's membrane. Because so Bruch's membrane is surprisingly tough and the tumor kind of inches through it and then is free to expand. The next thing that we often comment on is whether there's invasion of the retina. Yep. And then whether it's gone through here, the retinal surface or the neural layer and into the sclera. So those
Diane,
are all,
do you think there's a is there a retinal detachment here? How would we know?
Okay, so we can see that here is the choroid that's filling up with tumor, and here is an exudate. It's a pink exudate, so that means it has protein in it, and it's lying here between the choroid and the retina. So this is detaching the retina here, as well as the physical uplifting of the tumor. We'll talk next week about how we tell the difference between true and artifactual in processing retinal exudates. Okay, so I'm sure you've all heard of the calendar classification. So there's been some modifications to the calendar classification, and the current modification that we use in typing melanomas is whether they are spindle, mixed, or epithelioid. And if they're spindle, it doesn't matter whether we think they're A or B. We have to decide whether they're more than 90% spindle. So this one, okay, I'll just go back a bit, sorry. No, go back. Here we go. Right.
What does spindled mean?
Yeah, good question, Amanda. Thank you. Spindled means is that the cells are longer than they are wider. Okay, so these are spindled because they are longer than they are wider. Some of them are very much longer, but some of them are just a little bit longer.
How sort of sausage shaped, aren't they?
Yes, they are sausage shaped. So classically, spindle A had grooves and no nucleoli, but they're probably originally what we thought was spindle A cells are just remnant nevi. About a quarter of melanomas will have remnant nevi. And the spindle B cells are shorter, fatter sausages with nucleoli. So what constitutes an epithelioid cell and why are they called epithelioid? It's because the original pathologists who looked at them thought that they looked a bit like epithelial cells. So epithelial cells are a bit like poached eggs, right? So they're naturally kind of globular. And if you can imagine these in 3D, they're naturally kind of globular. They're round. They are not like sausages. And also epithelioid cells have big nucleoli in them. They're naturally more aggressive. Here we can see some epithelioid cells and some spindle B cells. So if you cut a sausage in half, you get a round profile. So how do I know that these little sausages cut in half are not epithelioid cells? Well, it's about the size and the amount of cytoplasm as well. So these epithelioid cells, I'm going to use another egg analogy, are like fried eggs with a whole lot of cytoplasm, whereas these spindle B cells, if you cut them in half, they have a much smaller cross-section, much less cytoplasm, and all around them you will see cells that look very similar but have ovoid nuclei. So if more than 90% of the cells are spindly, we call it a spindle type. If more than 90% of the cells are epithelioid, we call it an epithelioid type. And if it's in the middle, we call it mixed.
Diane, why do we care?
Because it's prognostically important. It's
good answer. Good to know.
All right. So team, in the chat, what is this? Spindle, epithelioid, or mixed? Amanda, what are they typing?
They're typing spindle.
Exactly. This one here.
More spindle.
More spindle, right? Spindle, yes. Great. And this spindle. One?
Just waiting. Epithelioid. Oh, you know what we should do to stop, what do you call it, a group think? They type and then but don't press enter, and then we tell you when to press enter, okay?
Okay, so this one, all right. We're going to, all right. So type now, what's this one?
But don't press enter until we say go. So type. All right, go. Press enter. Next.
All right, perfect.
That's
all
better.
Right. So what's the most important thing that pathologists report? You'll often see that people use a thing called the CAP protocol, which you can link to on the slide. So this is the College of American Pathologists protocol, which both Amanda and I like for reporting uveal melanomas. Okay, so most importantly, it tells you tumor cell type, the size of the tumor in contact with the sclera, so around the curve, the degree of extraocular extension, the tumor location, and then some other things that I'm going to explain in more detail. The complexity of extravascular matrix patterns and microvascular density, and together with these immunohistochemical stains. All right. So tricks in determining stage. So when examining an eye with uveal melanoma in it, you have to look very carefully at specific things. So the first thing to do is look at the scleral nerves and veins. And don't be fooled by pigment. Pigment naturally tracks along the scleral nerves and veins. You actually have to look for the melanoma cells. Look very carefully in the trabecular meshwork. Similarly, it can trap pigment, so look for cells. The ciliary body. Look at the iris for neovascularization. I'm going to spend some time on this next week, but that neovascularization can be a secondary effect of melanoma. And look very carefully at the optic nerves and meninges. So these will all influence the stage of the tumor. So what do we mean by extracellular matrix patterns? So this is a lovely epithelioid melanoma. And what it's doing is it's releasing lots of VEGF. And the VEGF is causing vascular proliferation. And this vascular proliferation is causing loops. See the big loop here? And there's a loop here, and a loop here, and a loop here, and heading off here, another loop. But here, there's no loops. So, okay, I would say that no extracellular matrix patterns are present.
Present.
Like you just have to say present in the tumor. Okay.
Or absent.
Or absent.
Diane, can you just have one loop and that's enough?
So technically you need 3 loops.
So this, I think, would be present. I think I can see 3.
Yeah. Okay. So we used to be really keen on reporting genetic abnormalities, and some labs are. So I'm sure that you've all heard about monosomy 3 being a bad prognostic factor and gain of 6p is a good prognostic factor. And then 8q is a bad prognostic factor in chromosome 3 normal tumors. But Amanda and I don't do karyotype anymore, because that's been replaced by doing immunohistochemistry for BAP1 and PRAME. Why do we do this? Because we can send the tumor off to the lab and get the BAP1 and PRAME back the next day, so it's faster, but also it's been shown to be prognostically more accurate. So what is BAP1? Has anyone heard of BAP1?
Also cheaper.
And also cheaper, that's true. So BAP1 is BRCA-associated protein, and it is on chromosome 3. And so it's a kind of surrogate for chromosome 3 loss. If BAP is lost, then it's going to be an aggressive melanoma. Can you remember what PRAME stands for, Amanda?
Preferentially expressed in melanoma. A clunky acronym, sort of, you know, some letters.
So
but
PRAME, if it's positive, means bad.
Bad.
Yeah, so we want BAP wild type PRAME negative tumors to have a good prognosis. So FISH, what's FISH? FISH basically counts the chromosomes. It looks like this. Okay, it can be quite technical. And this is the old classification where there was an initial GNAC mutation, and then it went down 2 possible mutations, mutation pathways, and BAP1 is where you detect chromosome 3 monosomy without actually having to do the FISH. All right, so in some very influential papers, what we are doing now is moving from the chromosomal division into 2 classes to the BAP1 PRAME division into 4 classes of prognosis for melanomas. So we've got the BAP1 retained PRAME negative tumors, which do very well. And then the BAP1 retained PRAME expressing tumors. And then we've got the ones that are abnormal for BAP1 but normal for PRAME. And then if they are both abnormal, then you can see that the survival is very poor. Okay, so that's both metastasis-free survival and disease-specific survival. The curves are similar. So often when we talk about BAP1, we will say whether it's retained, which is normal, or lost, which is abnormal. It's a nuclear stain. We're looking at brown. So there's a
It's
whole lot of
a bit confusing, isn't it, Diane? Because usually when we look at immunostains, you think, you know, the brown is what we're looking for. So,
the brown is kind of what we're looking for. Retained. So melanomas have got lots of brown in them. There's lots of brown from melanoma. So if the BAP1 is lost, it means the chromosome 3 is lost. That's the way I try and remember it.
because it's acting as a tumor suppressor, isn't it? So
it is. So yes, the BRCA associated protein does act as a tumor suppressor. Yep. So PRAME, right? So this is PRAME. PRAME here is expressed abnormally, and it's in various degrees. So there can be some PRAME staining or like no PRAME staining. So these are all negative, and this is various degrees of positive. So for the pathologists in the chat, you can score PRAME like you score ER, but even a low degree of expression is positive, like ER. I give it as counting it a bit like ER.
yeah, what's the baseline cutoff? It's pretty low, isn't it?
10% like ER.
Yeah.
10% moderate staining is positive.
Yep.
Yeah. Okay. So that brings me to the end of my chat about melanomas. Are there any questions about that?
Basal diameter, you showed some pictures where the globe was cut in a certain section, but what say you know, they're not, the base isn't completely spherical. What say it's like oval shaped and it's longer in the dimension that you didn't cut it in? Does that count?
You mean I'm getting a false short measurement?
Yes.
Yes, you do. We do have to be very careful to cut through to the largest basal diameter. You'll see if you go to the lab that we do many sections.
What about transillumination of the globe before you cut it?
That's also very useful for basal diameter. And just so I don't get tricked, I usually compare it to the MRI or ultrasound as well, because yeah, just yesterday I was looking at the basal diameter and going, that's too small, and I went to the block and I found they'd embedded it the wrong way around, without the maximum basal diameter at the top. And so today I've got them melting and flipping it so that I can see the correct basal diameter.
So why is that basal diameter so important?
It's for staging. It's for putting it into the prognostic groups.
Yep, stage. And Sarah asked, do you measure the curve of the basal diameter or do you straight line across?
The curve.
Yes.
Which can be, again, slightly tricky, but I usually do it like in increments.
Yeah. Or macroscopically, you can just roll your flat ruler around it, or you can use a piece of string. So, or a digital measuring tool. There's many ways.
Yeah.
How do you measure a ring melanoma?
Yeah. So basal diameter doesn't apply to iris melanomas.
Right.
Right. So good. So now we're on to the first case. So I'm going to go and I'm going to ask Amanda to put the first case in the chat.
No, we will train everybody to help themselves. Open your Google document.
Okay.
So no spoon feeding here. Feed your own spoons. Open your Google document and you will see the first slide picture, virtual pathology. And there's a picture of a enucleated globe, and it's got a little dent in the superior surface there. And how are we going to do a demo of what we do in the breakout rooms first, Diane? Because this will be their first experience.
It will. So I was going to put them 5 breakout rooms for
yes. Okay.
With a tutor in each breakout room.
Okay.
And so would you like to explain about
yes,
driving and things?
Yes. Well, why don't we do a demo ourselves on the
Okay.
first one. Okay, so I can open this up and I can be the driver.
All right.
And Diane will be the navigator. So I'm the driver, therefore I'm opening the slide and we're in a pair and I'm going to share screen. Just bringing that up now. Screen 2, share screen, share screen. So those of you playing at home should be able to see the same screen like this. And when I'm the driver, I'm using all my cognitive capacity to do driving and listening. But Diane is the navigator, so she's reading the map, looking in the textbook. She's doing most of the talking and I'm listening. So Diane, tell me, what should I do?
Okay, so the first thing that we're asked to do is to identify the normal tissue. So I just want you to stay still at the moment while we identify the low power features and the normal tissue. So I can see that the cornea is squashed in, and it isn't quite a transpupillary section, but the angles appear to be open, even though they're slightly squashed in. The lens looks normal. I can see the superior part of the eye here. Again, appears to be squashed in, which is artifactual. And as we move around, I can see that the retina is lifted off the sclera with a subretinal exudate. I can see that the optic nerve is looking pretty normal and likely not involved by the tumor. And then I can see this really large melanoma, which is present, extending from the ciliary body through to the equator of the eye.
How do you know it's melanoma?
Well, I know it's a melanoma because it's got brown pigment in it. And realistically, there's only one tumor that has brown pigment in it. Now, some melanomas in the eye don't, do they?
That's
fact,
right then.
There's a clone here that's growing in this melanoma that looks
Yeah.
amelanotic than the rest. I actually quite like it when they're amelanotic, they're easier to see.
So this has got dark chocolate on the outside and like white chocolate here.
Okay. All right, so I now have seen that at low power that there are some important things that I need to do. I need to classify it. So Amanda, can you zoom into the lighter area for me, please?
This bit over here. Okay, I'm just using the little scroll wheel on my mouse. Oh, it's an interesting pattern, isn't it?
So when I look at that at medium power, I'm seeing the vascular loops. So medium power, can you see the vascular loops, Amanda? You were pointing them out to me. So I put a big tick next to vascular loops. I can't see anything else interesting. I also note there's no necrosis and there's no hemorrhage. And let's go a bit closer again. All right, so I look here and I can see just at this power that most of these cells look stretched out. Most of them look to be spindle, but I also noticed there's some epithelioid. So just in the bit there, that's right, Amanda. Thank you, Amanda. We have some epithelioid cells. So looking around, I think the epithelioid cells make close to 10%. So I'm going to call this mixed. So a mixed uveal melanoma with extravascular pattern. So the next thing I want to do is I want to know whether it's through Bruch's membrane. So Amanda, can you go to the superior part of the melanoma, please?
Up here?
Yeah, that
or
bit.
At the edge bits?
Just where you think it might have poked through Bruch's membrane. All right,
Just
so we can see the retina overlying it, and the retina is quite attenuated over the top because of the pressure. Can we move to the left, please, Amanda?
This way? Anteriorly?
Yeah, anteriorly. Sorry, should have used anatomical things. I see that Bruch's membrane through there is looking fairly intact. It's that line, isn't it? So I would scoot along there and look to see if it had broken through Bruch's membrane, which it doesn't do.
Because it's not actually a mushroom shape, is it?
It's not.
Just
it's a big dome.
Big dome.
Yeah. Then the next thing I'd like you to do, please, Amanda, is I'd like you to go and look in the ciliary body. Because we have to determine whether or not there's invasion of the ciliary body and the angle. All right. So
Hmm.
is it in the ciliary body? There's lots of pigment in the ciliary body, isn't there?
There
So
is.
macrophages can contain pigment, but those don't look like macrophage cells to me because they have big, nasty nucleoli. So I think there is invasion of the ciliary body.
These are more epithelioid cells here
They are
in the
heavily-
ciliary body, not so spindled.
Invasion of the ciliary body. And then,
Mhm.
Amanda, can you please scoot along the sclera to see if there's anything that I
Okay.
can see there?
We might see some pigment, but you
Mm,
mentioned something about pigments in the emissary veins, not necessarily counting.
that's right. You actually got to see the cells. So I don't think we have any scleral invasion there. Oh,
Not
maybe.
so far. I'm
No.
just rendering, it's just coming up.
Okay.
I don't know. But pigment's not the same as a melanoma cell, is it? So
No, it's not.
there's pigment by itself there. There's a little bit of pigment out there. Do you think that'll count as melanoma?
I don't think so.
No.
Okay. So yeah. So in
Hmm.
summary, I think we've got a uveal melanoma, mixed spindle and epithelioid type, which has a basal diameter. Now we can estimate the basal diameter because we know the length of the eye to be about 23 millimeters, don't we? So it's going to be somewhere around like about 10 millimeters basal diameter, and more
See in
than
the bottom left, there's a little scale bar. Says 2 millimeters. Yeah. 2, 4, 6, 8, 10. Yep.
Yep, and then in height it's going to be perhaps 14 or 15 millimeters.
More.
Yep, and it invades the ciliary body. It does not invade the sclera.
Mm-hmm.
And then, and then we've identified the previous normal tissues. And then the question asks, what pathology tests would you do? And the pathology tests that I would like to nominate would be the Ki-67. So one thing we do with melanomas is we often count the mitoses in 40 hyper fields. Or we do a stain called Ki-67, which is used to identify cells in cell cycle. It's only good in the amelanotic bits. And a BAP1 and a PRAME. But those are the stains that I would nominate, and then that case would be complete. So the person who's doing the driving doesn't do the talking. The person who's doing the
Navigating.
navigate,
Does the talking, and then I'm going to stop share. So the navigator is free to look up all sorts of, internet resources, or it's really good to have your textbook on hand. You can flip that to the correct page and tell your driver, look over there or look over there. And it's more efficient to learn in a learning pair when you divide up the tasks. It's less cognitive load. When you go into the next breakout room, then you swap roles. So you're not driver all the time. You get to swap. And you'll probably have a different partner. So it's called promiscuous pair programming. And it's from computer science. And although we're doing this in a group situation, pair programming gets to better exam results as an individual than if you learn by yourself. That's why we do it.
All right, I'm just going to, I'm just sorting out the rooms now so that everybody has a tutor.
Oh, and we'll give instructions. So if you go back onto your Google document for today, Tuesday the 13th of February, and then scroll down to the next virtual slide link, which has been available all the time. The navigator will want to have open the how to talk about slides, make notes on A, normal tissue, B, tumour, low power, higher power prognostic, and E, what pathology investigations to do. So those are the questions to answer. And so we're going to look at the second slide, which looks similar but different in the next room. The how to choose the navigator and the driver. Actually, the person with the best and fastest internet connection is usually best as the driver to share screen. You just test it out amongst yourself.
All right, I'm going to open all rooms now. I'll be in room 3. So there's generally 2 or 3 people in each room plus a tutor, and we will see how we go with the next slide. All right, if you need help, then you can ask the host for help. You should just get a message to join. Matt, have you just popped out somewhere? Okay, and if you're watching this later, please take time to look at slide number 2.
Oh, I know. It's a big group.
Okay, so here we are back again. I hope you enjoyed that. Now Amanda and I, where's Amanda gone? Oh, there she is.
Yes, we're all back now. That
Amanda,
was fun.
now go through the slide, pointing out
Yes.
features.
And questions. So if there were questions that were unresolved in your session, please post them into the chat window now, and we will address your questions in particular.
Yep, there were some questions from my session, but I can remember what they are.
And yeah. Okay, and I've got the. So Diane, do you want to be the driver this time?
I will
So
be the driver.
Notice we're swapping the roles. And the first question is from Michael. How do we determine perineural invasion? And just wait till that share screen comes up.
Yeah, it's just having a little think. University of Leeds is having a little think for me.
It's all right. It's all right.
And also talking about some vascular loops again, please.
Mm-hmm.
This is what I'm getting from University of Leeds at the moment.
A grey screen.
Oh, there we go. Okay.
Yep, fine. It's loaded. Great.
All right, so some more on vascular loops and vascular networks.
And perineural invasion. Oh, perineural. Michael, would you want to elaborate on what you mean by perineural? Do you mean optic nerve or some other nerves? PNI.
Oh, just like if there's involvement of like scleral nerves.
Okay.
So if I was answering a question on how do you determine whether or not there is involvement of scleral nerves, I would always start with the macroscopic. So I would always macroscopically inspect the globe to see if there was any pigment on the outside of the globe in the region of common exits of nerves. And then at medium power, I would look to see if there was any pigment or purple cells tracking through the sclera. And then at high power, I would confirm that they are melanocytic nuclei around a nerve. So I'd be able to recognize a nerve by the wavy nerve nuclei. And then I would do multiple levels in that area to see how far through the sclera the nerve and tumor went. Because sometimes it goes right the way through the sclera and sometimes it goes part way through the sclera.
What does levels mean, Diane? You said you'd going to do levels?
I would do levels. So levels is a term that we use in histopathology to cut through the block that the eye is embedded in. So we normally have 4 micron sections, and what the people do when they're doing levels is they cut 4 microns and then they turn the machine that cuts the thin slices until they've cut 10 sections, and then they give me another section. So I get to see the melanoma every 40 microns, what it's doing.
How many sections could you get from a globe slice?
So they are 2 millimeters thick at least.
Lots.
So
yeah.
that means like 60.
Yeah, lots and lots. So what you're seeing is just one snapshot of a loaf of bread, one slice in the loaf of bread. There's more slices.
Did that answer the PNI question?
Yeah, thanks. Would you be able to just show what the nerve looks like, those nerve cells in the sclera, please?
I didn't see any on this with my group. Did anyone else?
No, I guess I would be looking through the canals, but I didn't see any either. It's a pretty rare type of invasion, I must say.
We'll Google it for you.
Perineural invasion be more important with cutaneous tumours like BCC, SCC, adenoid cystic, that sort of thing.
Yeah, but we'll find some. We'll definitely see it when we get to adenoid cystic.
Yep.
All right, you're going to tell me, Amanda.
Oh yes, what to do. We want to have a look at the vascular loop pattern. Perhaps we can have a look at a less pigmented area at medium power. Oh, okay. So I see, what do I see there? I see sort of like these loop shapes, loop, and then there's another loop. And like Bridget said, there's more than 3 back to back. So I think the vascular loop pattern is present.
Right, so this
rather
is
than
a loop.
Mm,
The pigmented macrophages tend to cluster around the vessels, which is why we're seeing it outlined. So
mm,
we've got 1 loop, 2 loop, 3 loops, 4 loops, 5 loops, probably
that's
6,
enough.
7 loops. So we have a vascular
Loop pattern.
vascular
So
loop
even
network.
if it's not across the whole tumor, does that count, Diane?
It does. It just has to be focal, though. I think it probably is.
Yep.
The whole tumor. Yeah.
Yeah. Next question, grading for size, AJCC versus COMS, C-O-M-S, Ella. Ella, can you elaborate what do you mean by COMS? We know what AJCC is, but I don't know what COMS is. Is that a clinical grading for size? And the other thing is, we shouldn't confuse grading with staging. So staging is how far it's spread, its extent, S for spread. Grading can be low grade, high grade. So
COMS is the Collaborative Ocular Melanoma Study.
So it's
is it a clinical or pathological framework?
It's clinical.
Okay, well, there we go. It's separate frameworks. They should correlate with each other, but you can't use a
I think
clinical
it's
framework
based
for
on,
that.
so I think it's based on using like at least ultrasound measurement. I wasn't sure if it was used also pathologically or not.
No, we don't use COMS. We use AJCC/tumor node metastasis, TNM.
Yeah.
We use a pathological staging system.
Staging. Yeah. And the grade is, of course, spindle mixed or epithelioid.
So not to confuse grade and stage. It's a common problem. How do we determine if it's just hemorrhagic or hemorrhagic necrosis?
Okay.
Let's have a look at some hemorrhagic areas. Yeah, there's a big lake of red stuff up there. I can see several of those. Oh,
Yeah.
What could that be? Looks like a sea of red with not much in it. So looks pretty fresh to me. What do you think?
Yeah, I think this is just hemorrhage because if it was dead tumor, I'd expect to see pink with like purple necrotic bits in, and maybe the ghosts of some structure. So if it's hemorrhage, it's just a sea of red. Where if it's necrosis, it's got
Dead cells.
and then dead cells. Yeah.
And this looks super fresh, so I think it's come out during the surgical manipulation in order to get it out.
These vessels are pretty fragile and leak.
Yeah, you've busted up some of those vascular loop networks and they've just bled into the tumour because they're very capillary sized. I don't see like hemosiderin either. So more questions? Oh, any role for SOX10, HMB45, generally these cases when it's obviously a melanoma, and that was asked by, would you stain it for anything for that? That Melvin's asking that question.
Oh, because I know it's a melanoma, I don't need to show that it's a melanoma because it's producing pigment. So it's
What say it wasn't producing pigment?
All right, so if it wasn't producing pigment, my differential diagnosis might include a metastasis from a poorly differentiated carcinoma if the patient had a history of one, for example. And in that case, if it was amelanotic and there was a possibility it would be a metastasis, then I would definitely do a SOX10.
Yes, I like SOX10 as well. What do you do, Bridget?
Yeah, we're doing SOX10 as a primary, or you might do Melanay or HMB45, but SOX10 is the go-to because it's such a nice clear nuclear stain.
Yeah, it's a nuclear stain, and we have we use ours with a red chromogen. So the trouble with the brown chromogen is the background melanoma, and you can't tell if it's melanin brown or if it is the chromogen. So we use a red chromogen for our immunohistochemical melanoma phenotype markers if it's amelanotic.
My group were asking, if we were writing the answer to this, they understand that they've got to write what's normal, describe the tumor, and describe all the prognostic factors, including its extent, its size, its grade, and whether it involves other structures. I understand that, but they said when it comes to what further pathological investigations would you do? Would we just write back frame P67, or do you think that they should write about doing fish for monosomy 3, 6 and 8?
Well, I think it depends on your laboratory. So you should do some prognostic markers, whatever is available in your laboratory. We, Diane and I, do BAP and PRAME, and so does Bridget. But if you don't have those available, which is highly unlikely, you could say fish or whatever, but some prognostic marker. What do you think, Diane?
I think I would probably answer it saying, what further investigations would you do? I would perform prognostic markers, BAP, PRAME, Ki67, and /or fish for monosomy 3, 6, 8q as available. I've just typed as available.
Because the thing that we're prompting for E is the prognostic markers, and then you have to list something. If you just say, I would do SOX10 and something that is so completely brown, you would not get the point.
No.
You would miss the point. You don't get any marks for that whatsoever.
Okay, do we have any further questions about this case in the chat, Amanda?
I think we have finished, I think. The last one I've got up there is how do we determine if it's hemorrhage or hemorrhagic necrosis?
Oh, again, we'd still see little bits of
we've done that one.
Tumour in it.
Okay. Yeah.
Okay, so I'm going to.
Oh, can you show us the tumour cells in the angle, please? Once it's just come
Oh
up.
yeah, I will do.
Henry.
Oh yeah.
Okay. Yes, and what about the trabecular meshwork? Oh, and there's 2 types of cells in the trabecular meshwork. And one of them is not melanoma cells. Oh, well, those look like melanoma cells. Look, there's some little dark small cells around about in the vicinity of the canal of Schlemm. They don't look the same as the melanoma cells, which are larger and have that nucleolus in the middle. So I think those ones there are the melanoma cells between the sieve part of the trabecula. And then outer towards the sclera, there's these dark fellows. What could they be?
Could they be lymphocytes?
That's what I thought they might be as well. So it's a trick, isn't it? They're not all cells.
Tumours attract lots of lymphocytes, and you will see us reporting whether there's a brisk or a non-brisk lymphocytic infiltrate in these melanomas. This one doesn't have many lymphocytes, so we'd call it non-brisk, but there are some lymphocytes there.
Tumour host response, right? So that's tumour cells in the angle over to the left, but more interestingly in the trabecular meshwork. Yeah, and one of those things is the canal of Schlemm, so very close to.
The canal often gets blood into it during an enucleation, so even though it doesn't have blood in it normally.
Yeah, so it's these things in the trabecular meshwork. Yeah. So, okay.
Okay, is there anything else that we need
Were there spindled cells anywhere, says Sarah?
All right, so I thought that this was probably pretty much all epithelioid.
Well, the answer is probably there are a few spindled cells.
These ones are spindle cells here.
But for classification, it has to be more than how many percent? More than 10% to be a mixed.
I think some of the areas have got some spindled cells in them. Sort of without, they're cells that don't necessarily have all the cytoplasm with them.
So when we're marking, we will take a clear photo of like a clear area. For you to write your exam on that. Yeah.
Diane, I think there was an area where there was some spindled cells that weren't lesional, and I think they were just little blood vessels or some sort of something else.
Right.
I think you were closer in them before, further towards the ciliary body. I think they were just sort of a big stretched out thin blood vessel, and I just wanted to make sure that everyone was clear on what was a lesional spindle cell versus a not lesional spindle cell.
Okay, so yes, I'm seeing here that we've got these are probably spindle cells here because they're much smaller than the cells. But then, yes, there's these things here, which could be some other cells.
There's also smooth muscle because we'll be in the ciliary body.
Okay, good.
Okay,
Right.
excellent.
I'll
How are we going?
let's stop
10
sharing.
minute break. We're doing
And-
good.
Yep. So we are back to
Break time.
break time. Okay. So we have got a 5-minute break, and I want you to all get up and stretch. I'm going to switch off the camera, come back in 5 minutes, and go make yourselves a cup of tea. It's a long afternoon, and we just need to work with our energy.
Oh, stop recording.
Remember to, yeah, sorry. You got me, Amanda.